Hello and welcome to this video on why the number and type of animals in research is important. This is an extension to the video Why Animal Research is Hard. Recently, a series of 3 European studies came out, and these looked at the effect and safety of GMO foods and glyphosate. This was a direct challenge to the retracted Seralini paper. They found that after 90 to 180 days, the rodents in their trials were unharmed. This is a significant argument against the proposed 2-year health studies of GMO food as proposed in Europe. Now, you may wonder why would this be necessary, and the answer lies back with that name, Seralini. He used a rodent study to justify concerns that GMO food and glyphosate cause cancer. The gist of which is that he took groups of 10 male and 10 female rats and fed them various GMO or glyphosate-laced foods. He then kept these rats for 2 years, and by the end of this time, many had developed tumors. This would sound damning until you know the details. This is also why many now fear GMO foods, as Seralini's study has had a lasting impact and its ghost has yet to be put to rest. Seralini used Sprague dawley rats. These are an especially genetically unstable species of rat, which has a very high probability of developing tumors after 12 months. Rather obviously, the longer you keep these rats in a research project, the more likely they are to develop tumors, and 24 months is simply so very far outside the window of time that is acceptable. Further to this, all of the animals develop tumors. This is unusual in most studies looking at this sort of situation. This is for complex reasons that will be explained later. This is also why the nature and number of research animals is important. Under normal circumstances, rodents and other animals can be a very good model of the human condition. This is despite common arguments that rodents make poor models, and you will find more details on that in the playlist linked in the top right. In this case, let's look at the Sprague dawley rat as an example of a species well suited to research within limitations. In this case, about 12 months. There are other examples, like the hairless rat, which has a compromised immune system and renal problems, both of which can very quickly complicate your research design. And then you have mice species and specific gene-edited lines. Of course, there are other species, cattle, pigs, sheep, rabbits, fish, frogs, and dogs being more common examples. Selecting the best model and strain is facilitated by services like PubMed's databases and other resources, or the NIH. Don't even begin to consider trials and research involving humans. The debacle with Wakefield and the number, selection, and later gerrymandering of the figures is a good example of everything wrong with the sample size and statistics of that study, and with most human studies. There is a reason why clinical trials are heavily regulated, undergo strenuous ethical testing, and are only then approved for limited numbers. Each species is used for reasons related to the applicability to the research project and desired outcomes. For example, you want to investigate the effect of effervescent products on the gut lining of an animal because you think that these things can hurt the human GI tract. You could use a pig. They have similar structures and dietary capacity. You can then feed these pigs a diet heavy in soft drink, Alka-Seltzer, and water. You then euthanize the animals, in many cases, or assess the damage through a proxy. This is because you can draw a comparable result in the pig to a human because of these similarities. You would not use a cow in this case, as it has the wrong GI tract when compared to a human. The data you draw is simply not consistent with the goals. By the same token, you could be looking at gene expression, in which case a rodent model with humanized genes would be better. Still, bear in mind that even with a humanized gene, there is a high probability that you cannot necessarily draw exactly the same conclusions. Much like with many other animal studies, this is more of a guideline than a rule, and you need appropriate numbers, which we discuss shortly. Returning to the rodent example though, Seralini used a rat species known to develop tumors later in life. This made the conclusion he drew untenable. The species he chose was inappropriate. If he had instead used a Wistar or Long Evans rat, the study would have been sound. The catch, of course, is that because these rats do not develop tumors, his conclusions would not have been possible. There are specific guidelines for animal research, and they are linked in the description. There is also a guide to selecting the correct strain. Things get even more complicated when you begin looking at different breeding facilities and strains. These have their own history and environment, which can affect the animal model. Many animal models begin from one research facility, and this is especially true of animal models. These start from one point and undergo a genetic bottleneck. In the case of some rat lines, this was intentional, and in others, has been the mutant offspring of these inbred lines. You will find that some rat species came about exactly because of this. Examples are the Wistar line of rats gave rise to both the Sprague dawley and the hairless rat. These differences affect more than just the genome. Different litters within the same facility will have different gut microbes, which can alter metabolism study details. This is true from animals of one facility, let alone between facilities of the same breeder, and therefore the same background stock, or on a larger scale, different backgrounds from different companies with different breeding lines. This is a variation within the same species, and makes drawing conclusions outside of it difficult at times, unless you allow for appropriate controls. This is why guidelines exist, as to the uses of animals in research. The number of animals in a research project is a direct product of these guidelines. The number of rats or other animals is a product of needing to reliably develop a predictive model based on an observed effect. This is also where Seralini's study gets stuck. He had 10 male and 10 female rats in each group. This is a critical factor in the failure of the design, which is only superseded by the timescale used. The number of animals dictates, or perhaps more accurately is dictated, by the need to achieve statistical certainty, if such a thing exists. By having a large enough pool of animals, you can adjust for chance occurrences. This is known as your sample size. This accounts for some of the effect size, although it is limited, and the chances of either a type 1 or a type 2 error. Ideally, you want a predictive power of 1 or close to, but this requires very large animal groups. Closer and more realistic numbers are around 0.9. The catch here is that many biological studies are at a power below 0.8, which is a consequence of having too few animals in each group. This is a common issue in neuroscience particularly. The consequence of these small groups is that they tend to overestimate any observed effect. It's like taking a room full of people and asking them if they like beer, wine, or spirits. If you pick a particular room, you'll get a very biased result, and this can be more pronounced, as a percentage, If the room has very few people present, this will naturally skew or bias your results one way or the other. There are calculators included in the description below that will help you if you wish to explore the idea of animal study numbers in more detail. So far, this has only really looked at the experimental group, but there is one more group that needs to be mentioned, the control group. This needs to be statistically powered to match the other groups you are comparing it to. And as with many studies, this is often underpowered, which means it's hard to see if there is a difference between your control and your experimental groups. This video has brought up the Seralini affair regularly, and that is for a simple reason. It was a high-profile, high-impact waste of a study. It failed in nearly every metric attributable to good animal research design, but like Wakefield, it has had long-lasting impact. This entire study was flawed from the start, based on the two major concepts that have been discussed in this video: the type of animal selected and the number of animals that were studied. Thank you for watching this video. If you found it useful, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.